And you go home and you eat the first apricot, which is lovely, and then you think, I'll have another one, and then you put the remaining 18 in your fridge. And in February of next year, I'll try and work out what all those things are at the back of the fridge that seem to be a sort of greeny grey colour. So things like shelf life are very important. Flavour, much more difficult because we all have a different perception of flavour. Designer products, and I do quite a lot of work in Malaysia, and there's a lot of interest there in manipulating oil, so it has different roles. At the moment it's primarily a cooking product, but there's no reason why you couldn't modify it so it isn't a product for engineering. Nutritional value, particularly colleagues in nutrition and food science are very interested in how we can modify a product so that it's got greater nutritional value, so that more of it is utilised by ourselves. I want to pick up on the first one, which is losses. So I've talked to you about the apricots. We've all got, I'm sure if we went into each other's kitchens, we've got a bin that looks a bit like that. And up to 50% of the crops that we produce actually are completely wasted. And if we want to contribute towards the global food security problem, one thing that we can do is say, cut out waste, to make sure everything we produce is actually used. And there are things associated with waste which are a problem, that as things get overripe, they decrease in quality, and they have reduced nutritional value. And if you're a farmer, that means lower income, particularly if you only get 10% or 20% of your crop to market. If you're a farmer growing mangoes in Africa or something, it may be only 20% gets to market. The rest actually rot in the field or when they're being transported. And that means there are fewer jobs because there's less money around, and that contributes as well. So there's this vicious cycle. People are hungry because there isn't the food, and also because the products are wasted. Now this is the time to sleep. I thought I'd do a little bit of biochemistry here. And some of you who are biochemists, I'm sorry if you spot any flaws in this. Those of you who aren't and you want to relax, now's the time. It's always good to have a break about halfway through. I'm going to talk about a biochemical pathway. And the biochemical pathway starts with A. A could be anything. It doesn't matter what it is. It's just a chemical. And in biological systems, often you have what are called linear pathways. So you start with A, and then as a result of a series of what are called enzymic reactions, it goes to B, and then goes to C, and goes to D. Okay. What happens if we stopped that final stage between C and D? Well, it's not difficult. We'd end up with C, wouldn't we? Because it would go to C and then stop. If we stopped between B and C, we would get to B and then stop. Or if we stopped at A, and if you're trying to drive through Sutton Bonington next week, that's exactly what will happen. You'll stop at about the crossroads and you'll get no further. That is as far as you can progress. Okay, nothing revolutionary there. Now I've given those letters colours for a good reason. Transfer those letters and those colours to a flower. Imagine that actually A is a white flower, B is a yellow flower, C is an orange flower, and D is a red flower. Now something I know that's troubled you for a long time and keeps you awake at night is, why are some flowers white and some flowers red and some flowers yellow, some flowers orange? Well, now we can actually work it out. It's because the biochemical pathway, which allows the pigments to be converted to different colours, are blocked. So white flowers are white because the next stage in the pathway has been blocked. And it's been blocked by a natural mutation. They're not genetically engineered. But what this tells us is, perhaps if we could understand how we could block those pathways by genetic manipulation, we could change the product. We could influence things. Can we do it? Well, we can. And this again was worked out here. I don't mean in the dining room, although the ideas may have come from the dining room. But in labs such as Don's labs and other labs around this campus, this was the first demonstration of what's called antisense technology. Talked about DNA. You're probably all familiar with the fact that DNA is broken into chunks, not physically, but in terms of how things operate, which are called genes. These are pieces of DNA which code for proteins. And normally what happens is that there is a piece of DNA which is actually copied into a message, and that message is then turned into protein. And if you want something analogous, it's a bit like a CD. You have a CD, but what you want is the music. Somewhere along the line, that information on the CD is turned into music. Well, here, the information in the DNA is turned into protein. That gene has a particular structure. And if you produce something which is a mirror image of that structure, you end up with something which is called an antisense gene. And that antisense gene, you could also fool the plant or animal into producing a message, and that message is the complete image of the sense message, and the two actually get caught up together. And when you do that, protein synthesis doesn't take place. So it means that potentially any linear event you could stop by introducing this antisense gene. Does it work? Yes, it does. If you have a look on the left-hand side, that's the normal tobacco plant, which is a red flower. The antisense one, what was done was to block the pigment synthesis process, and it was white. Now, flower colour is important, but probably not critical for food security. But I've talked about things like shelf life. And this is a picture of a tomato which I haven't actually manipulated, I haven't painted it, I haven't cut two tomatoes together and put them together. It's what naturally happens, but we've blocked the natural process. So tomato goes from green to red, and it's a linear sequence. And we blocked it by putting in a chemical, as it so happens. But the fact that it's a linear sequence means, can we actually block that progression? Because if we could enhance the shelf life. Now, fruit ripen in response to a gas. And probably many of you have ripened maybe an avocado by putting it with a banana. And the reason it works is because the banana produces a gas. It then gets into the avocado and causes that to ripen. Not all fruit ripen in this way, but many do. And that gas is ethylene. And ethylene is very important, and we've done a lot of work on this campus on ethylene. And ethylene literally is the catalyst, the trigger for causing these sorts of events to occur, and causes fruit to change colour and to have a particular flavour and aroma and change their texture. So having got that information was generated as a result of a series of biochemical projects. We needed to understand how the texture bit was regulated. Well, let's actually have a quick look at that. This is our green tomato. This is a fruit which is extremely unpalatable. It's hard, very astringent, doesn't have an aroma, and you wouldn't eat it. 2 or 3 days later, you end up with something like this, which is soft and tasty and has this lovely aroma. Actually, I don't like tomatoes. I don't know why I'm saying all this, but for those people who do, it's got all those characteristics. And the difference between that is really just sometimes a few days. The softening process, how does that actually work? Well, if we have a look at the cells which make up the green fruit, the cells are a bit like bricks. And the bricks are all mortared together. And if you take a wall, so these walls here, they're all mortared together, and if I went across and pushed it, even if I was really angry with the centre of the university, I still probably wouldn't manage to push that wall over. But if you take away all the cement and the mortar, the bricks have no strength at all. And then what happens is the wall can fall down. Well, that's why fruit ripens. When it's green and hard, all the cells are stuck together.